Hi, I'm Lauren Kirkland, and today I'm going to be talking about my final lab project for my Microbiology 214 class. And at the end of this quarter, we were given an assignment called Save the Patient. And in short, we all were giving an unknown organism, and we were required through lab testing and experiments to see if we could narrow down by testing to see what our unknown organism was. And so I'm going to be talking about that today. I'm going to be talking about the purpose, the why, the methodology behind it, my results, and just a quick discussion segment of it. So first, the purpose. Obviously, it was an assignment, but also to a big purpose and a why for me that could be used in the real world is I thought this was very relevant to me as a nursing student, as in a hospital setting or a clinical setting in the future. I could be presented with the same case study, right? Or be surrounded with this kind of same scenario. So in the case study I was given in this experiment, it was a patient came in with prolonged abdominal pain, diarrhea, and vomiting. So a blood sample was taken, results were we looked at those results, and then you start narrowing down, okay, what test fits this type of bacteria? And so that's what I'm going to be talking about today. And again, I think the big purpose and why is just that is very relevant. It's relevant to what I'm doing, what I'll be doing in my future, and it's relevant to what's going on right now in our world. So the 4 tests that I did to try to identify and isolate my unknown organism were the Gram stain, the catalase test, the blood hemolysis test, and the mannitol salt agar test. Now, we knew we had a group of 11 or 12 unknown organisms, right? And we were assigned 1. So a great starting place was the Gram stain test. And a Gram stain test determines what type of reaction the bacteria is going to have. The reagents used for the Gram stain test are crystal violet, iodine, your decolorizer, which is usually alcohol, and then safran. And when you add the crystal violet, this turns the bacteria purple. When you add the iodine, then that starts breaking away at the plasma membrane. And then your decolorizer, usually alcohol, for gram-positive organisms, it's going to stay purple. And then for gram-negative organisms, it's going to strip that color away. And then for your safran, for your gram-positive organisms, it still will remain purple, and for your gram-negative, it will be more of a pink-red color. So in my observation for this Gram stain test that I did first, I observed that I saw purple cocci. So I immediately knew I have a gram-positive organism that I'm dealing with. So that helped me tremendously in a starting place. I knew what next tests were relevant to gram-positive organisms. And so another thing that helps me too was my dichotomous key. So my dichotomous key, I have my 11 organisms up at the top. And then I knew, okay, the first test I did was my Gram stain. So I branched off into positive and negative. So I listed the positive organisms that I still had left, and then the negative that didn't apply anymore, right? Because I knew my organism, my bacteria, was gram positive. And another thing that helps me with this too, and that I can use in real life, in the real world, is the identification matrix. So this allowed me to see what type of probability, the probability of certain organisms combined with different tests would have. And so this was very relevant and often used in this activity as well. So now that I know what I'm dealing with, a gram-positive organism, I then think, okay, what tests are used for gram-positive organisms? And the first 3 that come to mind that we learned a lot about in this class and that we've done experiments on before is the catalase test, the blood hemolysis test, and the mannitol salt agar test. And so I just started with the catalase test. And I actually did all 3 of these in one day. And like I said, I knew this was a good starting point because I knew these tests were extremely relevant to gram-positive organisms. So the second test following the Gram stain was the catalase test. And the purpose of the catalase test is to see if my microbe had the enzyme catalase that was capable of destroying hydrogen peroxide. And if it destroyed hydrogen peroxide, that forms water and oxygen. And to perform this test, you inoculate from a culture and you streak on a sterile plate of your general purpose growth medium. And then you will put that plate in, you will incubate that plate in the 37 Celsius, and then you'll leave it there for 24 hours. Then you remove that. Then you will smear on a microscope slide, and then that is when you add your 3% of your hydrogen peroxide. And then that is when you're going to tell, is what I'm working with going to be catalase positive or catalase negative? And if it is catalase positive, then you'll see bubbles appear. As soon as you put that hydrogen peroxide on there, because that's the reagent used with the catalase test, you will see copious amounts of bubbles. If it's catalase negative, you won't see any bubbles. And that was my result. So my catalase test, it was negative. So then, not only have I narrowed it down from gram-positive organism, then I was able to narrow it down, okay, my catalase negative and positive, right? And so from 6, I had about 6 under my gram stain positive, and then my catalase test allowed me to narrow it down to about 3, and then I performed my blood hemolysis test from that. Now, I'm going to give you a list of still what I'm working with here. From the catalase test, it resulted in the negative organisms that I have are the Enterococcus avium, the Enterococcus faecalis, and then the Streptococcus sanguineus. Okay, so those are the ones, again, that have branched from my gram-positive, and then the catalase test, the negative result led them there. So now I'm working with, okay, what test can give me different results efficiently for these 3 types of organisms left? And I have 4 on the positive side that I'm not worrying about, okay? So then I'm doing my blood hemolysis test, okay? And the blood hemolysis test that I'm doing is I use the media, the 5% sheep's blood agar. And the purpose of this is to provide info on what type of hemolytic enzymes a bacteria possesses and whether or not they can destroy or destruct the blood cell, or if they can digest the hemoglobin. And so how you do this, how this test is performed, is you will streak for isolation on a blood agar plate, like I said. And in a clinical setting, oftentimes you can include many stabs of an inoculation into the agar to encourage anaerobic versions of enzymes to digest the blood cells, okay? But what I did was plate, okay? And you incubate this again for 37 degrees for 24 hours. And then the telltale signs are going to show you results of alpha hemolysis, beta hemolysis, or gamma hemolysis. And the result that I got was alpha hemolysis. And I saw a greenish color with colonies growing, right? So the greenish color and the colonies growing immediately told me that was an alpha hemolysis, okay? So if you were experimenting and, say, for instance, your results were an opaque transparent, that would be beta hemolysis. If there was no discoloration, it would be gamma, okay? So again, my blood hemolysis test led me alpha hemolysis. So then again, I branch on my dichotomous chart and I'm narrowed down even more. So my positive for alpha hemolysis, I have Enterococcus avium and then Streptococcus sanguineus. For my negative, I ruled out Enterococcus faecalis, okay? So I'm not worried about that anymore. So then from my alpha hemolysis positive branch, I then performed the mannitol salt agar test. Now in this, I'm getting 2 different results, or I'm seeing reflection in 2 different types of tests in one, okay? The mannitol salt agar test sees that the microbe can grow in high salt containing solution, and if the microbe can use mannitol as a carbon source, if it can ferment. And there's no reagents used in this. It's positive for growth in a high concentration of salt if you can see colonies. it's positive for fermentation if that red turns to a yellow. And how I did this was a tube to plate transfer. Again, incubating it for 37 degrees for 24 hours. And seeing the results of this, my results for this was no color change and no bacterial colonies seen. So I was negative for fermentation and negative for growth and a high concentration of salt. So then immediately I knew what my unknown organism was, and that was Streptococcus sanguineus. So on the positive side, it was Enterococcus avium. So immediately the mannitol salt agar, that last test I did, led me to my unknown organism being Streptococcus sanguineus. So that was very neat, and I was very pleased and proud of how my test that I selected gave me very straightforward answers. It was very efficient in narrowing it down each and every time. I didn't really run into any issues as far as contamination or anything of that aspect. However, I did have a little trouble in trying to identify my results for the mannitol salt agar and for the growth in the high salt solution. And I just had trouble identifying, was that a smear? Was that colonies grown? So I had a false positive the first time, but was changed to a negative. So that still led me again to my unknown organism being correct in Streptococcus sanguineus. Again, I thought that this was a very neat experiment that is very relevant to what's going on today. And very relevant to just day-to-day things that I will be surrounded in a hospital or clinical setting or anything like that moving forward in my career. And I just thought it was thought out well. It was challenging at the same time as rewarding, especially doing it in a virtual setting. I thought it was very neat and a great idea, and I still, even though it was in a virtual setting, like I said, have gained knowledge and feel like I've gained a lot of insight on what's to come in the future and how to be better prepared in relation to bacteria and organisms and testing and so forth. So again, thanks for watching, and this is Lauren Kirkland, and thanks.